Hello everyone, I'm Jianxu Chen from the Allen Institute for Cell Science. Here I'm going to talk about the method and tools that we have developed at the Allen Institute to obtain biologically accurate 3D cell and nuclear segmentation at scale. At the Allen Institute for Cell Science, we want to understand the cell. We believe we should be able to look at the cell and know what it's doing and what it did and what it will do. We believe this is possible because of the strong interplay between cell organization and function. Our goal is to create and understand a state space of cell signatures. We take a holistic approach considering all the main parts cell as a whole at the level of structures and organelles via 3D live cell imaging. To do this, we built the Allen Cell Explorer Toolkit, which includes everything from our stem cell lines and method to new simulation platforms. We want to pave an accessible and robust road from image to information in a quantitative and interpretable way. By solving our own challenges in order to move the science forward, we develop application-specific tools. It turns out that most of these tools generalize very well, so we hope to share this with others in an easy and accessible way. Today, I will focus on a key component of our cell image analyzer, which is 3D image segmentation. Here is an example of our standard cell integration assay for HiPS cells, in which we label GFV-tagged cells with dyes to be able to visualize the cell membrane and nucleus as a common reference system. So, for each image, we always have the cell membrane and nucleus channels, and also at least one more channel marking the GFV-tagged structure. It is mitochondria in this example. So, to analyze these images, we want to segment the cells and nuclei in all of our images so that we can analyze all different structures from a holistic view. This means over 18,000 Z-stacks from 25 different cell lines acquired in the past 3 years, during which many details may change, like the imaging system, the matrigel, and the dye, and so on. Let's look at Nucleus first. With the fast development of deep learning method, we are annotating 100 images in 3D and feed them into 3D UNet or 3D StarDist that can solve our problem. I know annotating 3D images are very time consuming, so can we just crowdsource the annotation job? We can take a closer look at a single Z slice. Here, it seems very easy to annotate, but how about here and here? Where is the boundary exactly? Even viewing from the side, like the side view here, it is still very hard. The boundary is very blurry in some sense. So in general, even though quite a few tools have been created, 3D annotation is just hard by nature due to the diffraction of light, lower resolution along Z, and for some more complicated morphology, like squeezing or folding, the annotation will be much harder. To overcome the difficulty in 3D annotation, our solution is to use specialized biological experiments to obtain more biologically correct ground truths for training. In specific, if we can label the nuclear envelope of the same cell as the DNA dye, here we just use our lemon B1 cell line. Then we can follow the nuclear envelope to inform us where is the boundary of nucleus. A shell-like structure will suffer much less from diffraction of light than the ball-shaped nucleus. Now, what about cells? With nuclei as seed, is 3D watershed accurate enough? We found that a simple watershed could be difficult to get accurate cell boundary. The key issue is that the top of the membrane is sometimes too dim for classic method to find an accurate boundary. Again, we use specialized biological experiment to obtain more biologically correct ground truths. In this case, we use the CAC cell line to inform us more accurate boundaries, especially near the top. We call this computation experiment co-design concept the training assay approach. The training assay approach is implemented with our Allen cell segmenter. We can use the classic part to create some preliminary results of lemon B1 segmentation and CAC segmentation. Then we feed that into iterative deep learning workflow, where the the preliminary results are curated with very minimum effort by either sorting or merging. Sorting is just like pass or fail, and merging is like circling out certain areas to use a different segmentation version. This is very helpful in dealing with cells in different stages. And then we use the curated training data to build a deep learning model. So here is an example of how we implement the training assay approach with Segmenter. We took some CAX segmentation result and used the sorting curation to get 7 out of 10, which are good results. Then train a CAX segmentation model with these 7 good images and apply the model on about 400 CAX images and use the results as the segmentation ground truths for membrane dye images. This will give us the final membrane segmentation model trained with the guidance from CAX. Okay, with the DNA segmentation model and the membrane segmentation model, we use the membrane model output to cut the DNA model output and then feed the result back at seed to get the cell segmentation. There are some extra steps mostly specific to our data and our analysis goals. The first one is we would like to consider the 2 parts in the late mitosis as one single instance or one single object. So we use an object detection model to fix the pair in instance identity. The second one is that in some very rare cases, the signal of the DNA dye and the membrane dye in certain cells are very dim, and the segmentation model may fail. We combine the label-free prediction from Brightfield with the segmentation model to further improve the robustness. And at the end, the method successfully scaled up to our cell integration assay pipeline, and we got about 216,000 cells from 18,000 field views from 25 different cell lines. With 98% of all the cells are well segmented, and 80% of all FOVs have entire FOV well segmented. This helps us making large-scale image-based single-cell analysis possible. By the end of this year, our segmenter will be updated with cell and nuclear segmentation code and trained models. More details will be on bioRxiv. At the end, we wish to thank our founder, Paul Allen, for his vision, encouragement, and support. Thank you.